Hello, everyone. Welcome to our monthly webinar on Target 2035. I'm Cheryl Arrowsmith from the University of Toronto, and I'm happy to introduce our hosts today. Matt Todd is a professor and chair of drug discovery in the School of Pharmacy at the University College London. He's a medicinal chemist with a strong interest in open science and open source drug discovery of new medicines. Currently, he leads several open source drug discovery programs in infectious disease. And Ben Perry is with Drugs for Neglected Diseases, DNDi, and he has a long history in this area of open science for drug discovery. And so I will turn the forum over to Matt, who will lead you through today. Matt?
Great, thank you so much. So yeah, this event is related to the Target 2035 initiative, and the speakers that we have today have enormous amounts of experience in various ways in securing molecules in open discovery projects. And so I think that we're going to learn quite a lot today from their experience. I'm just going to switch my screen on and run through a few things that I hope that we can cover today, hopefully. So let me just share my slides. Hopefully that is all clear. So the things I'd like to really talk about today, I mean, the theme is how we gain molecules for open discovery projects. I think everyone here may have a different opinion and view on that, whether we buy them, whether we make them in the community, whether we do both. So the background really is that the Target 2035 initiative will need a bunch of chemistry done. And the question is, where does that come from? So hopefully today we can learn a bit about what people have done in the past. So what has worked in terms of generated molecules that are useful for a research project, and what has led to the kind of online version of tumbleweed. Do things come from the public and private sectors, both, or one more than the other? How has the community operated? What were the rules? Can people get involved if they want to? Besides the molecules, have there been useful lessons in training people in how to do research productively? When molecules come forward, what kind of quality control do you need? If people give to projects, how are those contributions rewarded, particularly from ECRs? And generally, what might the speakers today, what advice might they have for the audience who are listening? Now, there's lots of people everyone has a different opinion about what open means, but I think all of us would be happy with the idea that open here means some kind of expanded, not normal collaboration network, maybe with some freely licensed asset at the end, I guess, as a general definition. What I don't want to talk about today, because we don't have time, and maybe we should have a separate session on this, is how we translate things that are open. So at the moment, I'm talking about molecules in early stage research projects, rather than moving through to the clinic. So our approach over the last 10 years or so is sort of summarized by these rules, which a few of you have seen before. Which were born out of an early project I'll mention in a moment. But essentially, we've gone by these so-called laws. It's like Lipinski, right? They're just suggestions, but they encapsulate how we go about projects, and really have been useful for clarity more than anything else. So the first three being a very useful way that people can engage with the project. Everything's shared, anyone can take part, no patents, and the other three are just about how to behave. And the way we've done that is to have lab books online that everyone can see, to-do lists being operated on something called GitHub, data being openly shared in things like Google Sheets, and a community based on social media. And the result of doing that, I guess, the social experiment has been that by making the project open, people spontaneously give, and they give high quality inputs. To summarize, what has been quite a lot of work over a number of years, ranging from students through to fully fledged professionals in the pharmaceutical industry. And the first project, which a few of you have seen before, was this one. And I just want to highlight this as an example of where things worked, I think, quite well, in that we required this molecule on the top left as a single enantiomer, working with the WHO. And the tumbleweed happened when we just sort of put everything in the public domain and hope for the best. But as we started to share more real data, and as we did a little bit of advertising, back in 2009, 2010, I think, on like LinkedIn, on a LinkedIn group of process chemists. We received a lot of spontaneous, wet lab experimental help, which accelerated the project. So that was something which worked well because we really tried to build a community around a specific objective. And in the end, we got to a nice solution for generating this molecule praziquantel as a single enantiomer with a very straightforward lab process, which has been taken on by both the generics industry and an international consortium called the Pediatric Praziquantel Consortium. That molecule is involved in a clinical trial in the Ivory Coast, but the thing I wanted to mention it because it worked in the sense that the project was definitely accelerated by our being open. And that required us to be very clear about what we couldn't do, I think. So since then, we've been working on a project called Open Source Malaria with the Medicinal Malaria venture. And again, there's a real mix of stories here about some things that worked and some things that didn't. It's hit to lead chemistry based on public domain compounds that were derived from pharmaceutical industry screens. And so, you need a bunch of chemistry done around things you start with in these sort of phenotypic screens. So the chemistry is sometimes quite early stage and exploratory, and in other cases, it's more sort of lead op chemistry. On the left there, we had a lot of early stage chemistry that was going on. We had quite a lot of freedom to operate, whereas on the right in series 4, you're starting with a fairly tightly defined core, which you need to explore. And so the chemistry is much more targeted and much more, I guess, expensive in that way. Early on, it's kind of interesting. I was thinking back to early on in the first series that we ran on a website called the Synaptic Leap back in the day. And there was a really classic tumbleweed moment where we wanted to try and work out what molecules we could buy and worked with a then small company called Enamine back in the day and other CROs to try to buy everything we could, which we did, and we got a bunch of inactives, but it was useful for the SAR. But there was a very amusing time when we reached out to a bunch of CROs and said, would you be happy to provide some compounds for free, pro bono, for this project, because it would be great advertising for you? And pretty much all the companies said, excuse me, what are you talking about? We're not going to do anything of the kind. Those that responded said that. So that was a classic sort of tumbleweed moment, expecting that I think these CROs would have the bandwidth or understand the nature of the open project. So that didn't work. But one example where it did work was where we had 3 molecules we needed to be made in the project and advertised the fact that we couldn't make them in the lab. And a student called Patrick Thompson, who was working, finishing up his PhD, whom we'd never met, of course, said, yeah, I can make that, and began to make it and sharing the data so we could see the quality. And then, finished off several compounds, got them evaluated, and became an author on the paper. A completely autonomous research project of several months, which progressed things and which led to authorship. So that's one example of a number over the years in open source malaria where things have worked really well, with a very low barrier to entry, I think, for people to manage their own research project. And then on the other end of the scale, there have been numerous cases of big inputs to the project from big pharma. For example, Scott Oback's lab and Pfizer doing biofunctionalization experiments of isolating metabolites so that we could test for potency. Again, pro bono contributions involving wet lab work, which advanced the science. Very, very helpful. And we've done quite a bit of work on crowdsourcing this with student cohorts. So we had quite a lot of success with student groups in universities, rather than just recrystallizing benzoic acid or something, would make new molecules as part of a live research project. And in the case of open source malaria series 4, we had this kind of hybrid route here where some things were made by a PhD student, like a stock of starting material or a difficult purification. And the rest of it could be done by students in the lab in a kind of crowdsourcing way. To train people, obviously, and to educate people, but also to generate molecules that are actually needed. So that's worked really nicely. Another example which hit the news much more was where we worked with some school kids to make a molecule called daraprim, that you may remember was the subject of a big price hike by Turing Pharmaceuticals. And there again, as far as we're concerned, it was an educational project that showed that with a great teacher, you could get new quality research inputs from a school lab. They just had a few benches and a fume hood, and they managed to do some really good chemistry there. We've since extended that in a project with DNDi that's trying to find new drugs for mycetoma, where a school in the UK is now making molecules as part of that project. And because it's an open project and you can see the lab, you can sort of see what they're doing. and how they're doing it. You know, there are really good training aspects to that. Even if you don't get a lot of molecules out at the end, there's great training aspects and community involvement aspects. So a lot of the people who've contributed, of course, were authors on this paper, and there have been papers since. And you know, authorship is a question. You have to have a discussion about who is going to be an author for which contribution and why. But it's very important that, you know, junior researchers who do make contributions to open projects like this do get the kind of reward at the end of being involved in a high quality research publication. So it's very important, even if the project lasts a few years, you don't forget anybody, and that people are rewarded in that way, and in a traditional way. Thankfully, open projects can be published now, in contrast to the early days when things that were in the public domain really didn't find an easy home at any journal. More recently, I guess we've had a lot of inputs from the private sector on a competition we ran to predict new molecules in one of the open source malaria series. And I guess this was something where we were quite surprised by the level of engagement by AI and machine learning companies in this project. And I like to think it's because the project's open, they could demonstrate the quality of the work they do. It created a problem for us because molecules needed to be made, and thankfully we had someone in the lab who could make the molecules. Ed here, who posted this. But the suggestions were really good, and the interaction, I guess, with the private sector in this open competition was really good. So of the entrants to this competition to generate predictive models, you can see that 5 were from the private sector, a very competitive area, but people were sort of happy to try and demonstrate their competence with an open data set. One thing we often say about open projects is that from a funded core, you can leverage inputs from others. And we've had a case study of this year in the second series of open source antibiotics, which was funded by something called Farm Alliance, which involves UNC Chapel Hill, UCL, and Monash. But outside of that, this sort of generates a lot of inputs from other organizations, which are shown on the bottom here, including DNDi in Northeastern, and companies like Haifa, who have done some metabolite work with us. So I guess this is a nice sort of discrete case study of a shorter project, which demonstrates that a funded kernel of activity can lead to inputs from lots of people. One place that didn't work was where we did an open source tuberculosis project with GSK, Triscantos. This is quite an interesting story because everything that we made in this very promising set of molecules, everything that we made needed to go through a check internally at GSK before it could be released. And it was about 2 or 3 months between making and releasing. And that essentially completely zeroed any sort of community involvement in that project. And so we never really built a community around that project, despite the fact the molecules are very potent and could, in some cases, be made in one step. It never really worked. It was really a GSK-us collaboration. We're trying a new project now. So a student at Sydney Uni is taking control of a new project in OSTB, which involves some of these cyclam molecules, actually breaking open the ring of the cyclam and having linear compounds. So I think we're going to see whether or not we can make this work in TB with a fully open project where we don't have that delay of revealing the molecules in the public domain. It is still quite easy, though. I mean, in open projects like this, it's quite easy to miss who is doing what. You know, if someone else somewhere else in the world is working on chemistry that's related to you, it's still quite easy to miss that. This example, I was just browsing through the waterfall of my Twitter feed and saw the molecules on the left that was being worked on by someone at the SGC, and I screeched to a halt because they look exactly like the molecules in Open Source Malaria Series 3. And so I reached out and we swapped molecules, and it was very productive, but I could have easily missed it. So we've, for a long time, thought that we need to make this little Twitter bot that notes what people are working on and tries to mutually, introduce them so that they can share things. Something automated, an automated sort of science introduction robot, or Cinder as a play on Tinder, so that you can sort of make these connections in a more sort of industrialized scale. But one of the useful things, I think, that, I don't know, as chemists, we tend to see things, then immediately that triggers a response in our mind to something. If you look at this molecule, like, many of you may be looking at that structure and thinking, well, actually, I know a molecule just like that. So I think there's still something useful in having a visual feed that can make connections between people. And we just started working on this, actually. So a very talented guy called Mike Robbins is making a little Twitter bot, which is going to randomly tweet out molecules and structures and links for open projects that require molecules or which are featuring molecules, just as a way of constantly advertising needs in case people see the molecule and think, oh, actually, I can make that, or that looks like something that I make. So we'll see if that works. It's an experiment in progress right now. So just coming back to the relevance to Target 2035. So the SGC is beginning an initiative called the Chemical Networks Project, which I'm going to help run. And the idea is that we do lots of synthetic chemistry. Where is that going to come from? So the aim of the project is to offer up SGC biology in return for chemistry from the community. That's the aim. And of course, we're talking about a very large scale project because there are lots and lots of probes that are needed in Target 2035. So we've been thinking quite a lot about how to structure that, and this slide's too busy to go into this, but the key message really is that if you're interested in getting involved in one of these things, or particularly running a project, so if you have a project you would like to convert to the open to generate a chemical probe for something, we'd love to talk to you about that, because I think we have a nice approach to this that we can clone for anyone's project. First one's just gone online, and we're about to put all the necessary FAQs and terms on the SGC website. But the first project is involved in this molecule here, which is has some nice data against a bunch of understudied chemical probes, understudied kinases, I'm sorry, and we just need to build in a little bit more selectivity for that. So we need to modify the structure a little bit based on a hit that came out of a Pfizer project. Just a quick, at the end, just a quick flag up for a possible resource that we might want to consider for samples. Many of us, of course, aren't interested in contributing, actual molecules to a physical repository of samples, because, for example, in the Chemical Probes Project, the private sector can provide probes that are needed. But should we want to get more out of molecules that are being made for open projects, there is the possibility of adding them to something called the EU Open Screen, where molecules are held and screened against a bunch of other things. A really interesting resource, which is getting going right now. So this is just really a plug for that molecules can be submitted and archived, physically archived, and then evaluated against a bunch of other things pro bono, for free, for people. So if you're interested in that, please do take a look. Just to thank some people and funders, I guess I do want to shout out to funders who are willing to fund open work like this. It does take a certain amount of courage to believe in the idea of this open mechanism that we're all familiar with will lead to improved science. And so I'm always grateful to people who are willing to put their dollars behind such projects. All right, that was just a summary from me, and I'll stop sharing so that people can ask questions if they would like, and we can move on.
Well, Matt, I see we have one question in the Q&A. So maybe I'll pose this to you. Have you considered using decentralized blockchain methods to incentivize private sector contribution by ensuring ownership through blockchain tokens?
So a couple of weeks ago, I wouldn't have known what that meant, but now I do. So yeah, I've been talking to someone about some of these ideas, and I guess we would have to have another session on blockchain and bitcoins and so on as a possible means of funding things. There's two things here. One is the idea of blockchain as a way of having being clear about what digital ownership is all about. So having a molecule which is maybe owned by someone, or having transparency in who's done what. That's always been less interesting to me, because I think we have a really good way of dealing with that with open projects, with open lab books and discussions and so on. It's clear who's done what and who's made which contribution. The second question, though, that's implicit here is about the funding potential of getting people to invest in open projects. And I think there is potentially something there, but that is going to require another session, I think, to get into that. 2 years ago, I would have said no, but now I'm saying maybe.
Okay, for the moment, I don't see any other questions in the Q&A. If anyone wants to ask questions, the Q&A is at the bottom next to the chat. I guess keep adding questions, particularly for Matt and others as we go through. We will try to respond to those that are there. If not, we will try to respond by typing. So I guess, Matt, if there's nothing else you want to add, I probably will switch across and tell you a little bit about what we've been doing at DNDi.
That's awesome, yeah.
Okay. Cool. Well, hang on. I've just seen a question pop up. How about this? How do these open approaches relate to open source in software? Maybe Matt, actually, that could be something you could handle.
Oh, sure. Yeah, there's always a lot of discussion about this. Some people don't like the use of the word open source for drug discovery because with open source software, the code can be losslessly copied. Whereas in an experimental project, you can't losslessly copy a molecule. So that is a distinction. I think that is a less important distinction than the similarity between the two approaches, which is that open source is describing a way of working, which means that you can get involved and the ownership is light and that you can accept contributions from others as the project is going on. And also that there are similarities in terms of licensing about your freedom to operate. So I think in that sense, there is a great deal of similarity in the kind of projects that I was just discussing and open source in software. Of course, we can't take something which I'm working on in the lab here and reproduce that immediately somewhere else in the world. But of course, we can share the information that's required to do that. And that would allow a molecule to be reproduced somewhere in as near as real time as we can make it. But I think really the similarities are strong in terms of how we work. And that's really what's meant by the term. Okay. Ben.
Okay, if anyone else has any other questions for Matt, please add them into the Q&A. Meanwhile, I'll share my screen and tell you a little about the work we've been doing at DNDi in this space. Let's have a look. Is that up and available? Okay. So yes, I'd like to talk a little bit about how we source compounds for drug discovery projects at DNDi, but I'm going to focus on two, let's say, innovative or interesting ways that we've been able to get molecules into projects over the last 5 or so years, as opposed to some of our more standard approaches. Some of them are quite close to the ideas that Matt just mentioned, but I'll just maybe a bit more of a deep dive on what we found in that space. First of all, a little word about DNDi, who we are. So we're a non-profit drug discovery organization, drug discovery and development organization. We're based in Switzerland, and we've been working since 2003 to deliver new medicines for a variety of NTDs for which, neglected tropical diseases, for which there is a huge unmet need. We also work in a few disease areas that are outside of the official neglected tropical disease space, like HIV and hepatitis C and malaria, more recently in COVID-19. So quite a broad spread. And as I mentioned, we're a not-for-profit organization, and we do almost all of our work, if not all of our work, through collaboration. And so we're very interested in this idea about open science and about how we can tap into the open science community to be able to push projects forward, because we try as much as possible to work with projects and produce products and medicines that are free from patent protection, IP-free space, that sort of thing, which open science really ties itself into. This is just a little snapshot of our portfolio, just to give you an idea of what we do and the size of our organization. So you can see that right now we have about 30 different products in clinical trials for a variety of different diseases. We're currently running huge trials in sub-Saharan Africa against COVID-19 across 15 different health ministries. A lot of work in leishmaniasis and Chagas disease, some successful late-stage products here for sleeping sickness. And we've launched in the last, since 2003, so in the last 17, 18 years, we've launched 9 new treatments for these diseases. Most of our early work is in kinetoplastid diseases. You can see here leishmaniasis, Chagas, and a little bit in sleeping sickness. So we have a very big discovery effort. Even within these little boxes, you'll see that there are many sort of sub-projects. I would say at any one time, we have maybe 20, 25 different active chemical series that we're investigating. So that's a lot of work. We're mainly funded to do this work by a combination of public and private donors, the various government organizations, Gates Foundation, Médecins Sans Frontières, and others. And that allows us to use what I would call like quite a powerful pharma-like approach when it comes to most of our drug discovery. So this question, where do we get our compounds from? Well, we have a classic model. Most of our projects run this way. We get money from funders and grants, and we work with the same all sorts of organizations that a pharma company would work with, mainly contract research organizations like Epicam in Australia, TCG in India, WuXi in China. We might also work with library vendors like Inamin and ChemDiv, and also collaborating labs, so academic groups who receive funding on grants to make molecules for us. We have active labs in India and Brazil and in the US. And in some cases, we also work with pharma companies who make molecules for us. This is great, but this doesn't really answer the question about how do you get open compounds from the open source space. So I'm going to tell you about two different ways that we've been looking at in the last 5 years for how to get molecules and where we can get molecules from. And as Matt alluded to at the beginning, there are different interpretations of what we mean by open. So one of these hopefully you will agree is very much aligned with Matt's view of open, truly open, everything can be seen, everything's visible. That's the open synthesis network here on the left. But on the right is more of an open innovation approach. So this is very, very closed. In fact, in some ways, very, very private way of working, very, very restricted. But it's a really innovative way for an organization like ours to access really interesting molecules that in theory could be applied to pretty much any project in the open if you could persuade various partners to participate. So I'm going to talk a little bit about this drug discovery booster project first. Just 1 or 2 slides to tell you about it, and then I'll move across to this open synthesis network. So the booster project is a collaboration between DNDi and up to 8 different pharma companies. I say up to 8 because Celgene, unfortunately, are no longer standing. They're now part of the Bristol Myers Squibb. So we're now back down to 7 organizations, but it's a collaboration in pre-competitive space using these pharma companies' libraries. So if you think about, most of these companies have been working for decades, making molecules for their oncology projects and their CNS projects and their cardiovascular projects. Most of those molecules end up in vials, which end up in dusty vaults, never to be seen again. But those, the nature of those structures are quite secretive because they reveal what those companies have done in the past decades. They give information about the sorts of targets they may have worked in. They don't really want to share that information with anyone. When you combine all the pharma companies and all of those hidden libraries, you have a real wealth of, in theory, available molecules just sitting there gathering dust. We were able to convince these partners, these organizations, to share those molecules with us in quite an interesting way. So the drug discovery booster is a virtual screening approach. It's ligand-based virtual screening. So what this means is we take a molecule, a structure that we found that's of interest for a project, and most of our projects are phenotypic, so interesting to us doesn't mean it hits target X, it means it kills bug Y. So we kill Leishmania, we kill Trypanosoma cruzi. We take a molecule and we show it to these guys, show it to these companies, and we say, what have you got in your collection that looks like this? And they go away and they use some pretty interesting complex computational tools to answer that question. Looks like or similarity is different. It's in the eye of the beholder and in the eye of the machine program that's used to do it. So we get different approaches to probing different libraries to answer that question. What's interesting is that we always get an answer to the question. We always get a list of about 100 compounds from each of these companies coming back saying, well, this is what we've got that looks like that. They share that with us. We screen those compounds and we get to see the structures. The structures aren't revealed to the entire organization, so they don't see each other's, we see them all. We test them and we generate data. Now, we only need a small sample to be able to do this, so we're effectively accessing 600, 700, 800 compounds at a time for free, testing them against our parasite and generating data, which we share back with them. And through a couple of different cycles, because this actually happens a couple of times, what we're allowed to do is, say, for example, AstraZeneca find one molecule in their collection that looks wonderful when we test it against the parasite, it gives us a boost in potency. Well, we can go back to AZ and say, this is a really cool result. Any chance we can show the others? And so almost always they'll say yes. We then show the other partners, look, this is what one of the partners came up with. It was the biggest improvement. Now that you have that information, do you have anything that looks like this guy? They go away and repeat. And we do these little cycles where they're sharing their libraries with us, they're sharing certain structures with each other, which is almost unheard of, and generating huge packets of data around starting points. And so we're able to put in what we call these seed molecules, and what we get out at the end are hit series, where molecules, very, very similar molecules within a series. So you can see we put in this guy, seed 2, and we get a lot of very similar molecules coming out. And hopefully what we're trying to find within this collection is something that improves against the starting point sufficiently to be able to put into an animal model of infection, which is the key readout that we're looking for our drug discovery projects to know, yeah, this is a series we really need to invest in. This has been very, very successful for us. So it's allowed us to tap into tens of thousands of molecules, proprietary molecules that sit on the shelves of these companies, not really available anywhere else. What we've learned is that there's very little overlap between these companies' libraries, so we very rarely see the same results coming back from these companies. We don't see many compounds that are available commercially, so it's different to commercial space. And we're able to drive our projects forward. So just to give an example, these are all of the different starting points that we've put into this project since 2015. It's about 22 different series. Most of these result in big changes. Most of these have resulted in improvements coming from these pharma companies' collections. And in fact, we've managed to get 6 different compounds put into animal models, or rather 6 different series, actually 10 compounds put into animal models just using this process. And one of those is actually kind of coming quite close to delivering a candidate for us. So this is a really interesting way for us to access chemical company collections in kind of an open innovation kind of space. Now we're able to do this because these are neglected tropical diseases. There's no commercial interest, so there's not really any competition or issues linked to these pharma companies sharing these molecules with us. Obviously, if we could break down those barriers that would then allow us to use these approaches in other diseases, that would be fantastic. But I think we're a little way off that yet. But we know that if we could, this would be really transformative. Shifting gears, moving to the other side of the way that we try, the other innovative ways we make our compounds is this idea of the open synthesis network. So this is very similar to some of the approaches that Matt just alluded to. It's tapping into universities to help deliver molecules. So the idea here is that, you know, when you're training as an organic chemist, synthetic organic chemist, which I am a synthetic organic chemist by training, you know, you've got to go in the lab, you've got to learn how to do reactions, you've got to learn how to do columns, you've got to learn, you know, how to stay safe in the lab. And usually you'll do that by following a tried and tested recipe that students have been in the same lab the year previous and the decade previous doing the same thing, making aspirin, acetylating ferrocene or whatever. Thing is, those techniques can be used to make molecules for drug discovery projects, right? There are plenty of molecules that are easy to make, quickly accessible compounds. I mean, Ed's going to talk about that later within the Moonshot. I think, you know, if you could somehow get students to make those molecules instead of all of them making aspirin, you know, everyone gets a little vial of white powder or off-white powder that then gets incinerated. Instead of that, have them all make analogs for drug discovery projects, which we can test and generate data. You know, that's kind of a win-win. It turns out, well, when we first started this project, we approached a couple of universities to see if they were interested, and we were quite surprised by the response. Pretty much everyone we spoke to thought this was, you know, a fantastic idea and wonderful. It's not novel. Other companies have thought about this in the past, but what enabled us to be able to do this quickly is that we were saying this is all going to be in the open. There's no commercial interest. We don't have to worry about IP. We don't have to sign over rights or anything like that. You know, we can just put the data out into the open. We can say, here are the molecules we'd like you to make. Here's the data that we use to inform that design. If you want to design the molecules instead using that data, go for it. Let's just make compounds. So that's kind of how this open synthesis network works. We have a list. We have a data set of molecules in a series with data. We have a list of molecules we'd like to make. The universities can look at that, they can add theoretical design molecules to the wanted list, and they can try to make wanted list molecules. Once they generate those, we'll ship them off to our labs, we test them to generate, in this case, usually anti-parasitic activity data, which we then share back with them and put back into the data sets. And so we have those sorts of cycles. There's a huge amount of enthusiasm for this. So this is what our network looks like right now. We started this in 2016. There are about 25 universities on here. We started off with some universities in the US, one in the UK, and a couple in India. And this is now growing to include universities in Brazil and in New Zealand and in Ghana and in Switzerland and Germany and France, or potentially France quite soon. And we've not really approached these universities ourselves. They found out about this somehow, and they've come to us and said, you know, we'd like to buy into this. So Matt was alluding to very similar approaches earlier, some phenomenal examples, you know, in Australia and with some of the partners that are on this list. I'm pretty certain if you really made a big push on this and went out and, you know, made a big deal and big splash and say, you know, this is a project that anyone can contribute to, we would be able to get hundreds of universities active in this. They are all very, very excited and interested in these sorts of approaches. We have all of our information on our website. So this is the open website that we're now using. You can see we currently have 6 active projects, actually 7 active projects. One of them is off the screenshot. In various diseases, so mycetoma, which Matt mentioned earlier, is a collaboration with Mycetos. We have a few in Chagas and a few in Leash. We have also some compounds being made for the COVID Moonshot that Ed is going to talk about in a little bit. Basically, we just put this on our website. Here's the molecules. This is where you'll find some background about it. Here's a list of molecules we'd love to make. Here's a list of data. Go for it. And here's an example of how that's kind of allowed us to impact on projects. This is the eagle-eyed amongst you will notice these are the same structures that Matt put up on one of his slides earlier, and molecules that came from this exact project are molecules that have now been passed on to the open source antibiotic project that Matt mentioned. But what we have here is we have a hit molecule here. You can see against Leishmania and T. cruzi. It wasn't particularly metabolically stable, so we weren't able to get this compound into an animal model to see if it worked. We actually initially used the booster consortium that I mentioned earlier to really elaborate around this. Doing that, we were able to get down to some pretty decent potencies, improve potency a little bit, maybe a little bit of improvement in the clearance, but not great. You can see this is a project timeline plot, right? So the earliest compounds here on the left, the later compounds here on the right. We initially did that booster work. This is potency against T. cruzi in this instance, colored by metabolic stability. So we're looking for something that's green and in the lower quadrant so that we can test it in an animal model. Nothing came out of the booster that was good enough for that. We then did some standard analoguing using a CRO making molecules, and we did okay. But it wasn't until we opened it up to the open synthesis network that we really started to make moves forward. So this is a whole series of compounds here that were made towards the end of the project by students at Imperial College and Northeastern working on this project. And we were able to identify compounds here that you can see we've boosted our T. cruzi potency down to sub-micromolar, down in the low hundreds of nanomolar, and improved microsomal stability. So we've been able to go from a hit all the way through to here using actually a combination in this case of the pharma company sourcing on the booster and then the university sourcing for molecules on the open synthesis network. Just to sort of show you how this is working in action, it's been a difficult or strange year for since the COVID-19 lockdowns have happened. A lot of labs have not been able to run their training labs. There's been a lot of universities completely closed, so activity has been a bit patchy. But just to give an example, this is activity we've had in the network in the past 2 weeks. It's not always this busy, but this is, I think, a decent snapshot. So you can see that we have a team at the University of Washington that I got an email 2 days ago from Kelly Kim, who's at the University of Washington, saying that some of her master's students doing master's thesis work on one of our projects have been able to crack the synthesis and get through to a final compound because we found the synthesis was really tricky. And that's going to be really interesting because we can share that information with some guys in London who are trying to make the same molecule and have been banging their head against the wall. So hopefully that will be a big breakthrough for that project. At the same time, we've got about 10, 15 new compounds made for the COVID Moonshot that Ed's going to talk about later from students in London and students at Williams College in Massachusetts. We've had the first draft of a paper on one of these projects coming out of the University of Nottingham. So as Matt mentioned, publication here is really important. We try to go for communal publication with as many people on there as possible, students as authors. And the first draft of this this particular T. cruzi project paper is coming has landed in my inbox last week. We've got 14 new compounds from the University of Munster on a Leishmania project, University of Beirut in Germany, who are very newcomers to this open synthesis. They just sent me an email saying, we've started working on this out of the blue. We've started working on these molecules. Where do we send them? Have made some compounds for Moonshot. Some students in University of Otago in New Zealand in an undergrad lab, 30 students in the lab at a time because they can do that in New Zealand at the moment, have everyone rammed into the fume hoods, making molecules for our Leash project. And we've even had new inquiries, from Curtin University in Perth and the University of Aarhus in Denmark, University of Lyon in France, asking, we've heard about this, any chance we can join? So it's starting to buzz. It's taken a while to get there, and there is a critical mass to be able to impact on projects. But like I said, if you really push on this, the amount of chemistry resource you can tap into is enormous. And this is all open, pro bono, in-kind, win-win situation work. So, just going back to this, as I mentioned, this is our classic way. I've shown you two other ways that we've accessed molecules. I should make a note, however, that both of these projects do receive a little bit of funding to be able to, well, in some cases, a lot of funding in order to be able to work. So the G8 Foundation, or the G8 Fund, sorry, in Japan, gives us most of the funding for this booster project, which is, a big project, requires a lot of logistic resource and a lot of screening resource. The Open Synthesis Network requires much lower funds, but we are funded partially by the Agence Française de Développement, so the French government, who are really interested in understanding this idea of open source science, open source drug discovery, and how we can do things differently. And I think that they're not alone in thinking that way. There are a lot of people, a lot of funding organizations who are starting to think about this. So maybe I'll open up to questions. I can see we have a few. Matt?
Awesome. Thank you, Ben. That's fantastic. I think we're just given time, that there have been really good questions in the Q&A, but given time, I might just move on to the next speaker. If you wanted to type some, I accidentally answered one of Frank Blundel's questions, so that's in the answered section, but I think he's got a particularly interesting question for you about how you organize projects like this as you start to scale up.
Okay, no problem.
That's all right. Okay, great.
Okay.
Yeah, fantastic. Okay. All right, great. So if you wouldn't mind just stopping your share screen, Ben. And we'll just move on to the next speaker, who is Lauren Webster from Dundee, and who I hope is going to tell us a little bit about drug discovery training for a resource-poor setting.
Thank you very much, Matt, for the introduction. Hope everybody can hear me. So my name is Lauren Webster. I'm based at the Wellcome Centre of Anti-Infective Research at the University of Dundee. And today I'm going to talk to you about drug discovery training for a resource-poor setting, as Matt kindly had introduced before. So if I could just move my slides on, there we go. So just a really short bit of background is what is the Wellcome Centre. So I'll be referring to it as WCARE, or as I become even lazier, CARE, throughout the rest of the talk. The Wellcome Centre, we take projects all the way from idea to preclinical status, where normally we partner with some industrial partners to take it further into clinical trials. What we do is that the Wellcome Centre was established in 2017, where we integrated an already established unit called the Drug Discovery Unit, as well as the mode of action teams in translational biology to help us on our journey to preclinical candidacy. So my role in the Wellcome Centre is that I am the training scientific and pedagogy lead. So what I wanted to do is today, I wanted to explain to you what we do in the training program, and in particular, what we do from the medicinal chemistry perspective, because after all, we are talking about molecules in this series. So for the training program is that we work with professionals, and I'll refer to them as trainees from here on in, is that we work with professionals from low and middle income countries to teach them how to undertake drug discovery. For the training program, that we have three main goals. The first is educational placements, where we will take these professionals and we will bring them to Dundee, where we teach them on how to do practical aspects in drug discovery. So we have dedicated trainers in biology, drug metabolism, pharmacokinetics, as well as medicinal chemistry. Moving on from there, we also deliver short courses in drug discovery where we teach these around the world. And as of recent, we also have some digital content that is freely accessible through our website, where the link is down at the bottom right-hand side of the screen. What you can see here is just a short animation running through on our how-to assay training series, which will be available on our website in the next coming months. And the last of our goals is engaging with partners from these low and middle income countries, such as Brazil, Ghana, Malawi, and South Africa. But for the purposes of today's talk, I wanted to talk to you more about our educational placements and what we do with them. So when a trainee comes to CARE, there are three different ways in which we can help them to deliver on their practical experience. The first is that we can let a trainee become embedded into one of our own projects. These are medicinal chemistry projects, but they are confidential. So we do work with industrial teams as well as grant holders as well. That means that the trainee may suffer from being able to disseminate their new knowledge to their colleagues back home. And the whole point of our training program is that we want to capacity build in these countries so that these trainees can come here, they can go back to their home country and teach everybody what exactly it is that they learned here at CARE and how for them to be able to replicate that, or at least to the best of their ability. What something is nice about the training project is that they are able to integrate themselves as part of our project teams. And I just wanted to highlight just this paper in ACS infectious diseases, where one of actually my first trainees, Rafael Vieira, who's based in Brazil, he actually contributed to this meta-RS paper that we published just last year. But it just shows that there was a large team that were involved in this, and Rafael was actually part of that, which was something nice. Because though we work with people from low middle income countries, that they tend to be working on natural product work and mainly in the isolation characterization. It is something that we do work with our trainees, but it's not something that we advertise as doing. And the reason being for that is because we look to try and make sure that we deliver training here that they can go back home and do at their home institution. And as people will know, that natural product work is it's lengthy, it requires lots and lots of analytical equipment as well as infrastructure that just perhaps some of our trainees lack at their home institution. But something today I wanted to highlight on, and I'm actually quite grateful that I follow one from Ben from the DNDi, because he's given a nice introduction to the open source network. So I'm not going to explain the ins and outs of behind the scenes bit of it, but I wanted to talk to you today about is what did I do with the open source network? So there is a large plethora of training that can be covered with just using the DNDi open source network. Something I wanted to highlight to you first before going through this list is that on the right hand side, we have two pictures taken from the same chemistry lab a year apart at the College of Medicine in Malawi. What you can maybe see is that these are chemistry labs, but they lack quite a lot of things that perhaps some of our own institutes have, the likes of fume cupboards, some shiny equipment floating around, and lots of people in white coats. These two images were taken from a training course that CARE ran with the College of Medicine, some purification, isolation techniques. And you can see here that on the bottom right-hand side, there are two participants here undertaking column chromatography, the equipment to which that we also donated to them. But you will also notice as well that there is a lack of their PPE. So they're not wearing safety specs. They also don't have any lab coats on. And that's because it's something that's just not available to them. So moving from this, we've set the scene on at least just one institution that we work with, but something in particular, I want you to focus in on this person here. You won't be able to see them very well in this image, but you will be able to in the future slides. So something that the DNDi open source network allows us to do is that we're able to tailor our training to every trainee that arrives with us at CARE. Now, some trainees may have some vast knowledge of working in the chemistry labs, some may have absolutely none. But what we can do is that we can make sure that we instill all these different learning points into their learnings. So what I wanted to highlight was the data analysis bit, is that I will be talking about that later on in the talk, but it's something that we really do like to focus in on with the DNDi work, as well as all of our other projects, obviously. But we make sure that all of our trainees leave here being able to analyze all of their own data. It's quality checked by us as trainers, but it's something that we get them to do on their own. We found that working with people from these resource poor countries is that they don't have these analytical equipments that even the basic of medicinal chemistry requires. But we did find out that, well, if we can teach them how to analyze the raw data, is it possible then that they can maybe work through collaborations of obtaining this raw data and analyzing it back at their own institute? And something that's really nice is, and the Ben also highlighted on it as well, is that it's a real life project with real life decisions. And I mean, I was obviously once a student and learning to make things in the lab, and to know that it just got thrown away at the end of the day, sometimes you do wonder, well, why bother? So this actually is quite a nice incentive to keep the student going and to ensure that also there's some form of delivery at the end. So it's not just about picking molecules and saying to the trainee, off you go and have a great time, please don't blow up the lab, there actually are an awful lot of things that the trainer has to do prior to a trainee arriving. What we like to do is that Ben also said to you that we are able to select molecules from the network, and we're also able to design molecules for the network and have them approved. And what we do as trainers is that we look at the synthetic routes that are available to us, and we look to see, are there any challenges in that synthesis? Is there something that is going to be good for the trainee's learning, or is there something that's going to be detrimental? And is it something that the trainer needs to take care of, or guide the trainee around it in some way to ensure that we still are able to deliver on our learning outcomes? But then you can turn this around and say, well, by the trainer doing these checks of the chemistry before they arrive, is that trainee advances, can we actually start to challenge the trainee? So there's actually an awful lot of learning that can be done just with one synthetic route. But there are things that we also have to remember as well, is that education is different between different countries. And in resource poor settings, this may become a bigger gap. So we also have to remember that it's the first time that some of our trainees are present inside the chemistry labs. And on the previous slide, you saw a chemistry lab in Malawi. And here is just a snapshot of a corner of one of our labs. They look very, very different. It can also be very overwhelming, actually, when you enter into our labs, not just because of the blinding light and the white background. But we also, as well, we can sometimes in this country take health and safety for granted. It's something that we think of to do because we've always done it. And it's something that's been instilled with us at such a really young, or not young age, because that means you're doing it as a child, but as in young academic age. And when we at CARE, we have very strict health and safeties, as most institutes will, but sometimes trainees don't have any of this health and safety information because they haven't been taught or know how to. So what we have to do is we have to make sure really early on that whatever chemistries we are doing, we have to make sure that they are safe to do, but it's also something that we're trying our best to see if they can replicate back home, or learnings can take them back home with them. Something I did want to point out as well is that every consumable may be a luxury, and there are pictures here of purple nitrile gloves. It's something that we've all been very familiar with. And I had a trainee one time, I saw him washing the gloves in the lab, and I asked him what he was doing, and he said, well, these have to see me through my entire placement. He was here for 10 months. And I said to him, oh, no. So you bin them if contaminated and you reuse. And he said, oh, I didn't think you actually really meant that. So that's something else that feeds back into this health and safety a bit as well. So we have to think carefully about how we start to structure some of these projects. So I wanted to highlight today is just one synthetic project that we undertook with the DNDi. I've been lucky to be with them since 2018. Enjoyed myself, so I have trainees in it, but I wanted just to highlight one trainee's journey through it. And this is Andrew Matewa. So the person I told you to remember on a slide just a couple of slides ago. This is Andrew. He comes from Malawi, and he undertook a synthetic program with DNDi and ourselves at CARE. Andrew had never set foot in a chemistry lab before. He'd never synthesized anything in his life, and what he had learned about chemistry was theory. So our first goal with him is that we said, okay, we would like you to make these 3 analogues. And for now, we're not going to tell you why we want you to make those 3 analogues, but we would like you to make them. We want to get you established, first of all, in the synthesis, and we'll start to fill in the gaps as we go along. So what we did with Andrew is we said, okay, here's a rough synthetic route. And now I would like you to try and figure out, how do you go from A to B? What are the sorts of equivalents you need to use? What is out there in the literature? Can you search through the literature? Can you start to tell me what's happening? And by doing this so early on, by not telling trainees everything, it gives them ownership at the very beginning, again, giving them incentive to continue on. Andrew, I have to say, did very, very, very well. And this is actually the final synthetic route that he did to make the 3 analogues that are outlined here on the right-hand side. I'm not going to go into details about how he found the synthetic route, about all the different routes that he did try, but I wanted to show you here that for somebody who'd never set foot in a chemistry lab, it was possible for him to do this synthesis. And there are different types of chemistries, which is something really, really nice about the DNDi network. It's not just amide couplings, it's not just SNARs. There are lots of different chemistries out there that you can do, or at least you can, if you want to design molecules, you can also create. So we've got, for instance, ring synthesis, we've got some Medicinal chemistry, deep protections, and couplings in final steps. Andrew just didn't do all these experiments once, he did them multiple times. He optimized, he used different temperatures, different pieces of equipment for reactions. He did all of that on his own. I merely was there to guide him, but it shows that somebody who had never made anything in his life and never set foot in a chemistry lab, it was possible for him to do this. How did we tailor this for his situation? So when he goes back to Malawi, well, we all do automated purifications. So we have combi flashes in our labs and mass directed auto preps. So we're very lucky to have them, something that which he doesn't have. Andrew did have to learn manual column chromatography. So to purify some of these materials, he did have to go through doing the manual column, something that students we've done previous and probably no longer do now. But Andrew also had the other added implication is that his institute and his surrounding area suffers from a lack of electricity. So there is electricity, but has a habit of going off every now and again. So we taught Andrew to do it in the absence of electricity, for example, the purification to a handheld pump. He probably hated me at the end of it, but it was very worthwhile for his training. But he learned how to do things that he could try and replicate back home in his home institute. It wasn't just about synthesizing molecules. So because, as Ben outlined in his previous talk, is that you can get given a data sheet, but it's not just about here's the data sheet, run forth with the suggestions on it. What we did here is we started to break down the data sheet, and this is where we started to do some extra education with our trainees, and we said, okay, looking at this data, let's start going through how do we actually start analyzing the data? How were those compounds that I gave you initially, how were they designed? How did I get there? And by providing us this data sheet that comes with lots and lots and lots of pieces of data on it, we're able to teach the trainees how to undertake the medicinal chemistry aspect of it. Synthesizing isn't just medicinal chemistry, it's organic synthesis. When triaging through lots and lots of data and including that in synthesis, that's medicinal chemistry. And how did we do that? So I did actually want to highlight about the software that we use to assist. The reason why I've got pictures here of Stardrop images, it's because Stardrop were actually really, really kind, and they've given me licenses that I can use for the trainees when they go back home to their home institutions. So at CARE, we do have funding that supports us to use this software. For the trainees, they don't, and Stardrop kindly donated that, so I thank them very much for that. So through the open source project, what were we able to do and what were the trainees able to achieve? The trainees find it much easier to be able to disseminate their knowledge to their peers when they go back home. They can say, look what I made and this is how I did it, something really, really good, and gets other people involved on the project. It can be tailored to a resource poor setting. I wouldn't say it's tailored for if you had to work in the dark and absolutely no electricity, but there is tailorings there that we've managed to circumvent use in our labs. I also hope that by showing you earlier is that there's more to learn than just making compounds, and this is where by having access to that data package, it really does enhance the trainees' knowledge and learnings on the medicinal chemistry subject matter. Without having to read through all of these bullet points, there are many, and I just had to pick a very few to show you today, is that it wasn't time dependent. DNDi didn't request that these compounds had to be made tomorrow, and with many of us working in chemistry projects, often we can be told the deadline was yesterday. You're like, oh God, keep going. So it was really nice that the trainees had time to be able to get a feeling for the project, feeling for the chemistry, and work at their pace. They can be challenged at any time, and they can perhaps speed up whatever it is that their training offers. But it's something that's really nice where it's not time critical. And something that may be asked for the future is that, okay, we've been able to do something like this at CARE, and Ben had rightly said, you know, we've been able to do it at lots of different universities, but can we actually do this in a low and middle income country? So how do we go about doing this? And I'm going to say just now that this is pending, as in, how do we actually set something like this up in somewhere that perhaps doesn't have access to a lot of infrastructure? I wanted just very, very quickly to say there are many people to acknowledge, and I'm not going to go through everybody by names, but it's thanks to the Wellcome Centre for funding for the training programme, to all of our collaborators, as well as our donators. And thank you very much to everybody for listening.
That is fascinating. Thank you so much, Lauren. We're bang on time, which is great. So you're perfect timing, and I'm going to use Chair's privilege to ask you a question, if that's all right.
Yeah, that's fine.
Because it's remarkable work. I want a thought experiment. What if you, what if you had a synthesis lab, like an automated synthesis platform, where everything was available in Dundee, and you worked with a chemist who remained in their own environment and did the synthesis remotely? Sort of says, okay, now we're going to column this, I want that spot, and they're going to interpret this NMR, and that's what it is. Would that, I'm assuming that you think that is less good because they're not actually getting their hands dirty. But so is it very important that the scientists are actually in the lab getting their hands dirty?
I would say from the perspective of our trainees at their home institutes, and as well as actually for everybody, chemists learn by doing. You know, to be told to do certain information, we don't learn, we switch off, we'll read an email or something like that. We like to be able to get our hands on something, and that's how we learn. We learn by mistakes. None of us can say that we got to where we were today because we had an unblemished career and we made everything that we intended to do first time. That's not the case. I mean, with Andrew's synthesis that he did, I let him do bits that I knew that probably wouldn't work, but he wanted to do it. He came up with very good evidence as to why he wanted to do it, and let him do it. And he did it. It didn't work, but he learned, and he learned not to do it again or do it a slightly different way. And I think if you get people to do this just virtually, you will miss that bit of their education. You'll miss that bit of the learning. And that's where our problem solving skills come in. As chemists, we are problem solvers. I think if you tell everybody exactly what it is that you wanted them to do and pick this spot, do that, there's too much micromanaging on that sense. And I don't think it'll be as worthwhile. You wouldn't put your heart and soul into it because you know that there's an aspect of you that you just don't have that ownership over it. And I think doing it the DNDi's way and how we've also tailored it with our trainees, I think gives them that incentive that yes. And that with Andrew as well, you know, I highlighted a very small bit. It's a lot of chemistry for him to do in a very short space of time. He was so good that we gave him even more stuff to do, and stuff that he picked on his own to do by using that data package. So I think you would miss that if you had that absolute control.
Yeah, no, that makes a lot of sense. All right, thank you so much. A question just popped up, which is quite interesting in the Q&A, if you wouldn't mind taking a look at that, but I guess we ought to move on. So thank you again. And then, so our fourth speaker today is Ed Griffin, who hopefully is going to tell us about compound sourcing in the COVID Moonshot project. Ed, if you're happy to take it away.
Thank you very much indeed, Matt. And I feel I'm going very much from one end to another. Just a second, I need to make sure this is shared right. Okay. So hopefully you can see my screen okay.
Looks good, thanks. Yeah.
Okay, I'm going to take you on a bit of a whistle-stop tour over the last 14 months of the COVID Moonshot project, which I will tell you about briefly in the first half of this talk. And then I'll make some reflections on compound sourcing, which is kind of the key point. I used to work in large pharma. I now work for a little spin-off company called Medchemica, and I volunteered for the COVID Moonshot in March last year. So what is the COVID Moonshot? It is the archetypal, informal, open source project that threw itself together last March. So Frank von Delft, near London from the Weizmann, and Alpha Lee from University of Cambridge came together when Frank became aware, working at the Diamond Group, that the critical main protease of SARS-CoV-2, the structure had been solved in China, but they wanted another structure. That the sequence was basically emailed to Diamond in Oxford. Group in Oxford managed to very rapidly reproduce that structure, and then the group at Diamond ran a fragment screen in under a month. I won't go into the full history, but it was incredibly fast. And then this was kind of advertised by tweet, which is one of the more innovative ways of kickstarting a project. So 60 structures were released on the 18th of March. Alpha and the group at a small company called Postera, which he just founded, created a website, which is here. Let me get my laser pointer going, which was here. And they basically put out a call to chemists across the world to say, if you design the compounds, we'll work out a way of making them, send them in. And that led to the creation of this really quite huge international team. So working around, so a fair few of us in the UK, but also critical folk in Israel, the Weizmann. Enamine stepped forward to help with the synthesis, and we'll talk about that. We've had compounds made in Wuxi and tested. We've had some inputs from SciLife Sciences, critical input from the IIBR, which is an institute in Israel as well. And then pretty much all the way across North America, we've had inputs from antiviral assays. UCB have been very generous in helping with design as well. So it's a great big team pulled itself together and started having designed compounds, getting them made. I'm going to take you through what's happened to give you a bit of context to when I talk about how we source those and the effect that's had. But the one probably major difference between this and almost any other project I've worked on, I've worked in some quite high pressure large pharma projects and biotech projects, is the absolute pace with which this has run. So I've never worked on a project that has attempted to run so fast. And that's been a real feature of it. And of course, you know, why? Well, because literally we're trying to do research in the middle of a pandemic, and every day you take longer is at the moment a couple of thousand more lives lost. So that focuses the mind more than somewhat. We're set off to try and find a drug, not a ligand, not a chemical probe, but an actual drug that could treat people, and a drug that could treat a lot of people very quickly. So if you think about the progress of COVID-19 as a disease, you basically have two parts to it. You have the viral part, where you get mild to moderate illness, mainly driven by heavy viral load. And then you get, if you're particularly unlucky and that develops into severe illness, this is basically the immune system getting out of hand, and you get severe illness moving into critical illness, and unfortunately in too many people, death. But in this point, once you're in hospital, the virus is essentially no longer driving the disease. So you've got to, if you want to really make an impact, you've got to go early. And if you're going to go early, that means you need to be an oral drug. It's got to be safe, cheap, and widely available. And you've got to start basically as soon as you can. You might even want to use it prophylactically when you've been exposed, so kind of in this bit before you're symptomatic, and you want to be able to reach the non-vaccinated population, and that whether those are people who are just vaccine hesitant or for health reasons aren't able to be vaccinated. But if you can flatten this, you flatten this, you reduce that, and we reduce the major load on people and healthcare systems, which is obviously the point. So one of the first things we did was, and it's very important, as soon as you think you're going to actually produce a drug, you want to say, well, what does our drug look like? So we spun up, and both Ben and I were involved very early in creating this target product profile as just what do we roughly think we have to be going at. We created this in the late March last year, really just taking what do you think a reasonable bet is. So you want to be active against the protease, you want to stop the virus, very simple PK/PD kind of relationship. Let's just get cover over killing the virus for 24 hours. We need spectrum, we need route of administration, we need solubility, and we need safety. And as of right now, we are in essentially 14 months from a standing start. We're beginning to get very close to our enzyme inhibition targets. We're closing in. I think this is not a good enough target. I think we need to be a bit more potent, but we're certainly getting to the levels of potency that we thought we might need a year ago. We have activity across spectrum of different variants, which is excellent. We have our current problem that we're trying to solve is all about oral exposure. Currently, we have too high a clearance. Solubility is not bad. Sometimes we get the right half-life, but it's not great yet. But super clean compounds, very good against a big panel of proteases, clean against a off-target panel. We have a bit of CYP activity, but we can mitigate it. So we're in a pretty good state. Let's sort of talk about compounds. So here's one of the series we looked at. And the reason I'm putting these up is these were all made, they're all made through UGI multi-component cyclizations. And although you can get down to around about 100 nanomolar, which is within spitting distance of what we wanted, the physical properties were really quite shocking of this series. So although we made, I think, probably hundreds, when we actually moved into looking at their metabolic profile, they were too highly metabolized, and we couldn't detach either the high log P or the metabolic clearance from the potency. So we couldn't get compounds that had both in any way. So we had to drop these very quickly, and that's the key probably difference to lots of the projects we've heard about so far, is we were being forced to make decisions extremely quickly. So if a series wasn't going to make it, we had to stop it and focus immediately. The second series we looked at came out of the fragment screening work that Frank von Delft's group led in Oxford. And I'm just going to show you a selection of small fragments bound into the main protease. So here we have 3 different fragments, all binding on to this histidine, forming a hydrogen bond, and then 2 different views of fragments that drop down into what is actually the P2 pocket of the protease. And here you can see all of them are overlaying. So this is just this view is just rotated, where you're dropping a halogen down into the bottom of this pocket. And well, as I said, part of the approach that was taken was to crowdsource the ideas. And one of the crowdsourced ideas when it was proposing, let's get these fragments joined up, was from Tryphon, who is also actually at the University of Oxford. He said, well, I've looked at all of these fragments. I can superimpose these by eye. And look, all you need to do is you join this bit here to this bit here, and that should work. Here's the conception. Starting materials are readily available. It's an aminopyridine plus a chlorophenyl acetic acid. Do that. So they did. And that works really well. So here's the crystal structure of it bound. So almost exactly as you'd hope. You can see here, there's the duplication of the pyridine. There's the chloride dumped down into the bottom of the S2. And when that was actually, we got the enzyme assay up and going, that came out at 24 micromolar. So from the first join So there was about 200 fragment joins were done at the start. We managed to get a 24 micromolar lead, and it's small and simple. And that was a huge boost to the project, because immediately you know you can make analogs around this. It's not a daft molecule. You can actually go to work. Over the next 6 months, so from that 24 micromolar compound, we explored what you could put in the S1 pocket, so develop that. So that brought us down, so that's about 30-fold. Then got another doubling by cyclizing around here. It turned out, very reassuringly, that all the potency lay in one chiral center, and you can see in the crystal structure here why that's very clearly, for want of a better term, a very chiral center. So we can get down to 140 nanomolar with this compound. And then as we were exploring the SAR, we found that just the addition of a methoxy group here gives you another 2-fold. That's a very useful compound. So now we're down in, this was about 6 months progress to get us an 80 nanomolar compound. Obviously, we have a number of analogs, and although the enzyme activity is important, the key thing is not the enzyme activity, but is actually the antiviral activity. So you can see here we've got a range of different analogs. Here's the oxygen, here's the nitrogen, here's the compound I just talked about with better potency. So against the VeroE6 antiviral assay, we've got fairly respectable potencies getting down to 0.5 micromolar. Calu3, which is a different cell line, plaque forming assay, we've got really good separation between the cytotoxic effect and the inhibition of viral growth. And here we're down at getting on just below 300 nanomolar. And if you want a really good visual picture of what that means, so this is taking this compound on the right and looking at different strains and comparison to remdesivir. So this is the South African variant, which is quite resistant. You can see in terms of remdesivir, even at 2 micromolar, you're getting very strong breakthrough. This compound is just a different naming of this. You see wipeout at 2 micromolar, slight hints at 1 micromolar. Yes, you get breakthrough.
I don't think, as Alan says, I don't think deciding, I think moving it down to America.
Okay. Somebody's keen to share.
That was Al chipping in, don't worry.
Yeah, great. So as you can see, we have good antiviral activity. The challenge is not enough oral exposure. So, you know, the compound is relatively highly cleared, although we see visibility, IV and oral. Even if you're going at 100 mg per kg, we're not getting enough cover. So this is our current area of focus in terms of medicinal chemistry. We have pushed the potency even further forward. So our latest compounds, we're now down, given these are racemates, we'd be expecting these to be down at 25 nanomolar. This is an overlay of 2 X-ray structures where you can see we're starting to possibly pick up an interaction here. There's the previous best compound, which was 84 nanomolar resolved. So we're making some significant progress. But the other things I'd point out are not only are we making progress in terms of potency, in particular, we're improving the physicochemical properties. So although we're making more potent compounds at lower log P, and that makes a great difference when you're worried about metabolism and solubility. The assay cascade is really stripped down to actually answer the questions we're working on. So we have enzyme, rodent cassette, solubility, potentially efficacy. Pretty much everything else gets kicked down. We have antiviral assays going on the side. Okay, that's given you a real overview, and I'm probably slightly overrunning, and then I get to the meat of the project in terms of what I was asked to talk about. But I'd say our needs very much defined our approach. So we had to do custom fragment linking, and we're in a hurry, so we bought FTE chemistry. We're doing expansion around our leads to get potency. We needed generic library chemistry to start with. We wanted a big supply of commercial building blocks. So we went to a big building block supplier to do that, which is enamine. We are doing some very bespoke, challenging cores at the moment to solve our PK problems. Again, that's being done with FTE chemistry. We have done a lot of nanoscale chemistry actually for rapid hit expansion. That's been predominantly done at the Weizmann, making hundreds of compounds in plates. As I said, solving our solubility and clearance issues, classic lead optimization, it's all been FTE chemistry. And what I wanted to kind of present as an idea is there's actually a tension between medicinal chemistry and synthetic chemistry that we might as well face up to. And so I trained as a synthetic chemist, spent a decade of my life doing it. But as a medicinal chemist, what I really want is I want test results bluntly, and I want them now. I want actually as small a molecule as possible. Particularly in the case of the COVID moonshot, we want compounds as soon as possible, and we want them in the right quantities, maybe low milligrams for enzyme, but we'll need up to 300 plus milligrams when we're going into efficacy models. Purity, usually you want good pure compounds if you're going into cellular assays, and you want critical impurities removed. And that doesn't just mean nasties from synthesis, that can mean active impurities because you're modifying a compound that you know is already active. And so if that's a contaminant, you get inaccurate results. And we've had cases of that. And clearly we care about form, so solids and formats. So we need our compounds plated out, ready to be pushed through to assays. And often the chemistry is bluntly quite dull. We want those variants on a theme. So we need access to a broad range of building blocks. And when we want resynthesis, we want it now because we have an assay for it to go into. And sometimes that can be a challenge for some academic synthetic chemists because these are different needs. And often people are interested in demonstrating a particular method, solving a problem, making a complicated molecule. And that's really well suited to making complex chemical biology tools and early stage process improvement. But there's clearly a tension. And one of the concepts that can lead to is this tension between over design and under design. So we've had a huge number of people contribute ideas to us, but some have clearly an overestimate in their ability to predict potency, solubility, or clearance. And we all do this, but it can be dramatic when people over design molecules because they make the molecule too complicated because they're trying to fit every single hydrogen bond or be very creative, make unusual ring sizes or kind of whatever. But that makes molecules slow to make. And you make too few to understand the SAR. There is also under design, of course, which is if you don't even think about what the SAR is already, and we put all our SAR out into the public domain, you can see all of it. And then people make molecules with poor physical properties as well, because they don't do a simple C log P calculation, or make insufficiently diverse compounds because they're convinced that they know just the one it should be. Bluntly, that often leads to either inactivity, insolubility, and particularly lack of cell permeability when you get the phys props poor. But there is what I've described as Goldilocks design, so not under, not over. And this is just an example taken from a couple of months back from Matt Robinson, who works in the project, works for Postera. And this is his entire design rationale. Very simple library probing new position of N in the ring. So in other words, we'd moved from an oxygen here to nitrogen here, and Matt proposed 5 compounds, and methyl and sulfonyl, the acetamide, the NH urea here, and the unsubstituted urea. 5 compounds. Requested 28th February, shipped 17th of March. And we get a really good spread of data because, the log P's in square brackets, so from 2 micromolar down to 200 nanomolar. But if you notice, in terms of log P, this is brilliant because the more polar compound, is more potent, and that's exactly the kind of thing you want. And of course, Matt did have a hypothesis that he was hoping that he'd be able to pick up some interaction out here, and it looks like we have. But the follow-up to that is, because we believe that that's picking up that glycine 189 interaction, is then a very small set of follow-up libraries. This is about 40 compounds in all, with adequate diversity. So yes, sulfonamides, but not obsessing about them. Some sulfonylureas, more amides like this, because that's not a bad potency. Another urea, some carbamates, and not abandoning the idea of alkylating on here by reductive amination, and even some heterocycles. So this is what was proposed. And
hey Ed, sorry, I totally don't want to rush you, but I'm going to have to slightly rush you. I'm so sorry.
No, I will go as quick as I can. It worked a treat, and the key learning is that actually don't abandon a poorer bit of SAR because it can get you to what you want, right? And it was shipped in a month. The last 3 slides I have, you can buy so many good compounds now for so little money. And if I was running a project where I was just lead hunting, I'd just go to Enamine and I'd buy myself a bag of compounds, because you cannot beat this in terms of price. We do synthetic route prediction on every molecule. Here's one of those. We use Postera's tool, which they made available to us, which is super cool. You put the molecule in, it tells you what building blocks are available and a route. Get in touch with them. This is one of the backbone tools of the COVID Moonshot, and it works a treat. Right, here's, I'll just fire through it. You've probably read it by now. We have made 1,300 compounds with 140 FTE months of effort in the last year, which is crazy fast. We have had some really good synthetic academic inputs, mainly around nanoscale, but most of it has been FTE library based. If you are a synthetic chemist on this call, please get in touch with me, because we have stuff for you. We have problems. We need to scale up our molecules because we're going to need multi-kilo amounts, and we will really appreciate your input. Contact me directly here, or direct message the COVID Moonshot on Twitter. That's how we work. I apologize for overrunning, but think about, when you're choosing how to source your molecules, what are you trying to get? Because that should determine the approach you take. And if you're going all the way to a drug, lead optimization is going to sink a huge amount of FTE resource, and that isn't news to anybody. If you do join us, you end up on the paper that's already being read. We have 131 co-authors and growing by the day. Sorry for overrunning. It's kind of a bit of a frantic project. Thanks very much for your time.
That's awesome. Thank you so much. Really, really fascinating. I'm sure there will be lots of questions about what you said. So if people could put those in the Q&A, that would be great. But in the interest of time, I'm going to have to move on. Thank you so much, Ed. So next week, I think Ben is going to chair the next couple of talks, but I think the next talk is going to be from Bill Scott. He's going to be telling us about some of the crowdsourcing he's been doing over the last few years. Looks good, Bill. Great.
Well, thank you. So I'll tell you first a brief introduction to distributed drug discovery, which is going to be our approach to an open source distributed synthesis of molecules that could be valuable in medicinal chemistry. The concept was built around the idea. I came from Lilly, many years of research in the pharmaceutical business, but I also was very interested in education all that time. Came to IUPUI. What I wanted to do is combine education with a real serious application to a research need. So what I thought was, if we could focus our students on ultimately coming up with drugs to treat either neglected or infectious diseases, we could have them simultaneously learn all the critical skills that are needed in the medicinal chemistry process. And this just kind of shows you some of them. But the idea was to show them the integration required in a discovery process, and then motivate them in the process of trying to find these drugs to treat really serious needs. realizing that in an educational framework, we didn't have the resources that we would have at a place like Lilly or Merck or Pfizer. We took both a combinatorial approach and a distributed approach to problem solving. So the idea in a very simple form here was, if at each stage of drug discovery, I just illustrate a few of them here, we could take a big problem, break it into small chunks, and distribute it to schools throughout the world, where we would involve students in learning that basic skill at the same time they'd be solving a small chunk of the problem. So I'm going to focus on chemical synthesis here, but you can imagine the distributed process also working at all these other levels of activity in drug discovery. So the challenge was, how are we going to get undergraduate chemists who have never done any synthesis before to make molecules that have significant promise of biological activity? And we took this combinatorial approach, by the way, also solid phase based, because again, working with undergraduates in a complex synthetic procedure, we need to have something that minimizes the opportunity for loss of material. Solid phase synthesis is just totally appropriate for that. We developed over a number of years, we started this program in 2003, these combinatorially based solid phase procedures, and just showing you some of the scaffolds that could come out of them based on fairly simple chemistry. The first example being alkylation reactions that introduce unnatural residues, which will become unnatural amino acids, which we then acylate. But all these procedures, if we're going to consider them D3 procedures, have to be reproducible, have to have been shown to be doable by optimally multiple students within our school, but also at global locations. And the way we did that was to take some of these procedures and take them to other schools and show that we can make the same molecules using those same procedures. So reproducibility was a key component of the distributed drug discovery process. I just want to show you that what looks like a very simple molecule here, in fact, if you enumerate a catalog based on the alkylating agents that we know from both our experience or from the literature will work in substitution there, and then take a handful, 100 carboxylic acids, and then just show you the structures. These are quite complicated structures to expect an undergraduate to make, but they can make them. Obviously, these are very interesting and highly functionalized molecules with a very simple procedure. Another aspect which has been brought up, I think Lauren talked about this, the critical need, if we're going to distribute this in an educational framework, to have a very inexpensive process. So we focus all our work on having things being simple, inexpensive, powerful, and reproducible. This is the equipment the students use to do the combinatorial solid phase synthesis based on those various procedures that I showed in the previous slide. The core of this is a very simple glass tube with a glass frit in it, and that's all you need as a reaction vessel for solid phase synthesis. Mounted in a grid here, students can do up to 6 reactions at a time. They can wash the beads. They can finally collect their products in vials after they cleave the final product from the beads. Just to show you the international aspect of D3, that first procedure, the D3 Lab 1, where we make unnatural amino acids on resin and then acylate them, we took that procedure and that equipment to Moscow, Lublin, Poland initially, and showed that the students there, also in Barcelona, could make the same set of 8 to 15 molecules in pretty much the same purity at all these different locations, just following the written procedure that had been worked out by our students here, a represented crew of those students at IUPUI. But recently we took it to the University of Havana. We've taken it to Puerto Rico, Mexico City. Again, the core driving force being showing that students anywhere in the world can use these procedures to make pretty interesting molecules. So I'm going to just show you now the application of that to a particular project within D3. Here we had found through some student work where they were making unnatural amino acids in an undergraduate lab, we had identified, well, really rediscovered a series of fluorinated phenylalanines that were quite active in inhibiting the growth of Pseudomonas aeruginosa. It was either the fluorine at the 2, 3, 4, or 3,4 difluoro position. All those molecules were quite active. Well, we went and did a literature search after the fact and found out that a number of these already existed, had been shown to be active against Pseudomonas aeruginosa and probably not pursued as drugs because subsequent literature showed that they had toxicity in mammals, presumably because of the resemblance of these fluorinated phenylalanines, the phenylalanine itself, and that thereby inserting the fluorinated phenylalanines in a normal metabolic process to produce abnormal results. So we then set about a project to see if we could deal with that toxicity at an undergraduate level in undergraduate labs. And so one of our first highly distributed programs was to make prodrug derivatives of 5. The idea being, if we have unnatural or natural amino acids at the C terminus of a dipeptide containing one of the 4 different fluorinated phenylalanines, maybe we can, if we search through these and test them, identify a subset that is still susceptible to cleavage by a bacterial peptidase, but not by mammalian peptidase. And in that way, within the cell of the bacteria, have it degraded back into its suicidal product, the fluorinated phenylalanine, but protect our cells by virtue of not having that same peptidase floating around in our bloodstream to produce the toxic fluorinated phenylalanine. So we set about as our first target then these unnatural amino acids, and this is where we distributed. We had it done at our school at IUPUI, Colorado College, Goshen College in Indiana, also UIndy, but also Santa Clara University on the West Coast, University of Havana in Cuba, and then University of Puerto Rico and UNAM in Mexico City. These folks doing some trial runs on that basic reaction sequence. And the reaction sequence that we gave to our folks was one of the simplest ones that we could come out with to first explore these aspects. So rather than exploring the unnatural amino acids, which we have done subsequently, and is simple again by the route that I showed you in the D3 Lab 1, we just did this, what amounts to a dipeptide synthesis, but now done on resin beads and capped with various fluorinated phenylalanines. Well, how does this result then in a series of molecules that would be appropriate for an open source distributed problem solving? If you look at the structures that would come out of that simple sequence, remember we have 4 different fluorinated phenylalanines, the 2, the 3, the 4, and the 3,4-difluorophenylalanine. We can construct computationally this grid of 80 compounds from the 20 different amino acids for each one of those fluorinated phenylalanines to get all 80 of these possible combinations. So we constructed that grid, and then we worked with our collaborating institutions, be them small, where they just make 6 molecules, or be them IUPUI, where we've had up to 160 undergraduates doing this kind of research in their regular undergraduate lab. We don't make 160 molecules. Again, reproducibility is really critical. So we have students make, a number of students make the same molecule. So we may have 4, 5, 6 different replicates of the same compound, 24 as an example, done by the students. We always have a control molecule. So it takes the laboratory into the mode of a typical undergraduate lab where we know what should be made and they can characterize it. But at the same time, the students make a new molecule. We have yet to make the 19 molecules in red. To kind of summarize, using that as an example, to summarize the results of this process, in this case, we established these productive 8-school global collaborations. We educated students while they reproducibly and globally synthesized multiple new potential drug molecules. These molecules were shipped to us or to be tested at IUPUI in a, by the way, in a biology lab that was, again, in a kind of D3 mode, something that students could do. So that was part of the regular series in microbiology. Yeah, that was where the lab was. And so they would test the molecules then in turn that the students made here. And then we also sent these to the community for open antimicrobial drug discovery, which for free would test these against a panel of very serious either fungi or bacteria. Based on that, the result of that and another collaboration, which I don't talk about here, was that we end up discovering some potent molecules against Pseudomonas aeruginosa and in addition Cryptococcus neoformans and Candida albicans. We did patent those. Now that's always going to be subject to controversy, and I'd love more input on this. And I don't think this is a resolved issue yet, but our institution really was interested in getting some IP, and whether or not that was something we want to do in the future, and how that will complicate an open source discovery project when the molecules aren't made by just IUPUI, but all our collaborators in a replicated fashion, are going to be challenges. What are some of the challenges that we are facing now? We are a small operation. It's just 3 of us really who are leading this project. And so there are the practical aspects of compound tracking, storage and handling, managing the test capacity. Right now, if we look at just the Pseudomonas aeruginosa example, we're the ones doing that testing. Our students are doing the testing, but as more of our collaborators are making molecules, it's becoming unmanageable for us. So in the distributed drug discovery theme and mantra, we want to work with our collaborators to help them establish their own testing tools for these molecules. So the cycle can be done within the institution of both synthesis and testing. We right now have a project which we're heavily involved in COVID against the main protease, Ed. So we've just made 70 odd molecules from our undergraduates, which are close analogs of some of the published inhibitors of main protease, but we don't have that implemented at IUPUI, the protease assay that our undergraduates can do so that they can test the molecules. So that's an immediate challenge for us. And we want to distribute testing. Informatics obviously is always an issue, capturing, sharing, and analyzing this data. I mentioned the ambiguity, and I know there are many opinions in this area. I believe it's still not a settled issue, because when we think forward about how these things are going to be taken through clinical trials and the inducements that we need to have, it's not clear that intellectual property won't be an important part of that equation. Funding, here we sell this project on the basis of simplicity, lack of expense. In fact, point of fact, the institutions are funding a lot of this themselves, because they're just placing this into the regular undergraduate program, either in an undergraduate lab or as independent research students. So that works pretty well. However, Marty, myself, and Gino are self-financing this project to develop the tools that are then implemented in these institutions. And that's only going to last so far. We are overloaded as IUPUI coordinators, and then we are also getting old, frankly. And so the question is, how are we going to establish some sustainable program leadership for a program that we think we've demonstrated pretty well works in multiple institutions and is deserving of further continuity. Just a few comments on what we might suggest to facilitate maximum productivity of these global contributions. We would love to participate in others' informatics and computational analysis platforms. We want to be able to figure out ways to further minimize the demands that this program will place on our undergraduate educational collaborators. As you know, these folks are just totally overwhelmed with the work that they have to do. It's tough sometimes to get them to replace or splice into a laboratory that's already going as work for years, a new procedure that will involve a little bit of tweaking and understanding. And so we want to try to make it as simple as possible for them to splice this in without upsetting all the other initiatives and load that they have. Part of this also involves creating a reward system that encourages academics to participate in ways that aren't normally acknowledged. So, this is open source. The ways we published a number of these papers with the multiple collaborators, and we need to figure out ways to make sure they are fully acknowledged for what they do. Facilitating local institutional funding. Again, in the mantra of distributed drug discovery, in the best of all possible worlds, the funding itself is distributed. And we've had a number of examples now where the institutions themselves, under the umbrella of running a new D3 lab, have been able to get funding internally to make that thing happen, to buy the simple equipment that's required to do this, to get the chemicals. Again, I mentioned the sustainable leadership is a real issue for us, and you know, part of my desire to be part of this program is to point that out and see if we get some help on that. Jen, I don't know how many of you are aware of this.
And then a structure.
Yeah, I don't know how many of you are aware of this initiative, Just One Giant Lab. We recently became aware of it, well, probably within the last 3 or 4 months. But this is an intriguing platform that may help us deal with a lot of the not outsourcing, but problem solving at all these different levels through a community sourced challenge. And with that, I will complete my presentation and be open to questions.
Thank you, Bill. That is great. Really fascinating program. I'm gonna have to move on to the final speaker to make sure we get done in time. But there's already a question from Ben in the chat window. Panelists can't put questions in the Q&A for some reason, so it's in the chat window. And I'm going to ask you one as well in a moment. And I'd encourage everyone else to ask questions in the Q&A if they'd like to ask Bill about this. It's a fantastic program. But in the interest of time, because we've only got about 15 minutes left, and I want to make sure that Paul has enough time, I'll ask Paul to switch on video and share slides and give us the last talk of the day.
Thank you, Matt. Enjoyed hearing those. It's a pleasure to join this group. I'm probably the first biologist to speak, so shifting gears in many ways, or in a couple of ways. So much of this talk is going to be about the non-profit chemical probes portal. Let me just get my pointer working. There we are. Telling you about this non-profit resource, which I've been closely involved with from the outset, actually in 2015, and is very closely linked to Target 35, both in terms of the objective of getting a probe for every protein in the human proteome, and also in terms of the people involved. And in addition, I'll just show you a couple of examples of collaborative projects that our group's been involved with, one of which is a completely open source project, and the other is an example of academic and commercial collaboration. This is my disclosures, and I think most relevant to this is my role as now the director of the nonprofit portal, which is located or operated out of ICR now in London, but is a truly international nonprofit project, the purpose of which is to help biologists select and use suitable chemical tools to produce the correct answers to the questions they're asking about the role of proteins in biology and disease. So just a little bit of background for maybe those of a more chemistry background to see what the real issue is facing the use of chemical probes in biology and in disease and drug discovery research. So like the Grail Knight in the Indiana Jones movie, biologists need to choose chemical probes wisely, otherwise it's very easy to get garbage from very reproducible but completely meaningless or incorrect results if you use the wrong chemical tool. And for some time, chemical biologists and drug discovery people have been preaching to the choir, celebrating the development of best practice and the development of potent and selective chemical probes, but oftentimes the biology community that needs to use these and wants to use these reagents is not hearing the messages because they read different journals, for example. It's for that reason Julian Blagg and I, my then very close chemistry collaborator, published this paper, which I think was the first review type paper on chemical probes to be published in a biological journal as distinct from a chemical biology or a drug discovery journal. And the messages that we were trying to get out to the biology community is, what's the best practice for using chemical tools to get the correct answers in biology? To have them think about, and at least be aware of the fact that there are some good chemical probes, there are some bad compounds, and there are some truly ugly molecules that continue to be published in high-profile biology journals where the conclusions made is completely erroneous, meaningless, and can waste time and resources, and even allow drugs to proceed to the clinic that have the incorrect mechanism of action. And so we're talking about, obviously, pains compounds. So you chemists can see all of these types of structures that are truly horrible, that biologists not uncommonly use in the output of screens and are published in important journals to claim a mechanism-based effect, which is quite incorrect. Or it could just be that you have a chemical tool which is pretty good and might hit, say, one kinase, or might hit several kinases, and that's okay as long as you're aware of the properties of that molecule. Let's just look at one example that chemists here and even the biologists might find really startling. It was published in Science Translational Medicine by the Sheltzer Lab. They'd published one article before this, but this is one where they took 10 drugs that are actually in clinical development, each reportedly acting on a specific protein. They CRISPR knocked out the gene responsible so that the protein, and then selected cells that no longer had that protein. They showed that in contrast to the previous genetic experiments and previous work with chemical inhibitors, that all of the protein targets that were claimed for these compounds were non-essential. Sorry, I should have said this is a cancer context. So none of the targets that were claimed for these compounds were essential for cancer cell growth. That makes you wonder about what the compounds are doing. Moreover, here's another clever experiment. In those cells from which you've removed the target, if you test the compounds, the drug efficacy, the cell killing, was equally active in compounds that completely lacked the claimed ostensible target. So these compounds are killing cancer cells through off-target effects. So it shows the value of stringent genetic validation and mechanism of action in any biology, actually. And it applies to chemical probes as well as drugs. Chemical probes are there to show the effects of a particular protein in a particular biology or disease process. So they need to be selective. So I think this validation, which is showing a loss of activity in CRISPR-KO cells or cells with resistant mutants, and if you're a chemist, then you can work with biologists to achieve these types of cells. Technology is becoming increasingly straightforward. It does raise the bar for chemical probes and drugs. It's not essential in the first stages, but it's added to the list of biological characterization that needs to be done if you produce a chemical probe. So biochemical action on the intended target is necessary, but these examples show often not sufficient. So how to choose your chemical probes wisely? I think it's really important that chemists as well as biologists understand these kind of rules, and the whole thing is helped by multiple resources like the Chemical Probes Portal, which is an expert advice resource, Probe Miner, which is more computational, Drugs and Probes, another very valuable resource, and of course Structural Genomics Consortium and the Donated Probes, Chemical Probes project, and others that you've heard about. So these are very simple messages, but surprisingly not well understood in the biology community outside of drug discovery. You need to be aware of the requirements of, and I would say that chemists need to be aware, if you're going to make a chemical probe, you need to be aware of what the biologists are looking for at the end of the day in a chemical tool. You need to be aware of the requirements of target, the assays that you've got, and the biological system. What are you trying to achieve? And if you want to find the best compounds to do your experiment, then use expert curated resources, use computational resources alongside that, which can be complementary. And absolutely avoid using historically biased publications or Google or the vendor catalogs, which will sell you every chemical probe that's been published, with oftentimes no information that allows you to make a selection. So you need to be absolutely critical about the quality and the limitations of the probes that you're going to use. You need to consider probe availability and costs, and you need ideally to use more than one chemotype, so 2 different chemical classes giving the same effect and controls, and using best practice procedures. And these factors need to be considered in probe design by the chemist, as well as what the biologist will, how the biologist will eventually use those tools. So there are some useful guidance. There's a publication that my colleague Ian Collins and I put out in 2010, which I'm not going to go through this in detail, but you can see various chemical requirements, potency requirements, selectivity requirements, and biological context requirements. We call these fitness factors. You can reduce that down to a smaller sort of at a glance list, and this is the SGC criteria. So less than 100 nanomolar potency for the target, at least 30-fold selectivity for proteins in the same family, profiling as much as you can against other families, for example, kinome profiling and pharmacology panel profiling if you can afford that, on-target activity at ideally less than a micromolar, and use alongside orthogonal active chemotypes and inactive controls. So these are the types of, although the portal doesn't require these absolute, as it were, quantitative factors, reviews for the portal use these figures oftentimes. Although a pathfinder compound, I should say, that doesn't yet fulfill these criteria, if you're en route to achieving the best possible chemical probe, pathfinder compounds en route to this can be valuable. So we need to recognize that. And Cheryl and Al, who are on the call, have been key players in this from the beginning. It's important to stress that drugs and probes are different in that, of course, drugs have to be effective and safe. Chemical probes have to be selective for the protein, oftentimes more selective for the desired protein than the target. So it's not as if sometimes people think, well, a probe is en route to the drug. That may be true, but often the probe needs to be much more selective than the drugs. And there are even issues around chemical probes in this interesting paper from Batya Shapira and colleagues. So we're accelerating the pace. I think Cheryl introduced at the beginning, target is to get by 2035 probes for the entire proteome. We've come a long way from the first guidance, which is Phil Cohen's advice on how to use kinase inhibitors and how selective they were and so on. And I was involved in that in the early pharma collaborations that worked with Phil on that. You know, we're really accelerating here, and it's a community effort. The numbers of publications per year with chemical probes in the abstract or the title, it's similar slope, is growing exponentially, which is great. However, as my colleague Albert Antolin published in his ProbeMiner paper, less than 5% of human proteins have even minimal quality, let alone best practice chemical probe criteria. So need to speed up, need to increase quality of the compounds that are used as chemical probes and the ones that are used by biologists. We need to get the message out on best practice to the biology community. I think we all have a responsibility to do that. And both chemists and biologists have a crucial role to play in this community effort. So if you're not familiar with the chemical probes portal, go take a look. Here's the website. I'll tell you about the new website in a second. It's an open online resource. The idea is to change the way biologists find and use high quality small molecule reagents for their experiments. It's aimed to make it easier for our experts to select the right chemical probe or the best chemical probe they can to initiate their study and to help them use and design a more informative experiment. It's established alongside the Arrowsmith, Cheryl's paper with a big bunch of authors in the field. In 2015, it's now been highly cited. It's based on expert recommendations and commentary on selected probes. It provides an an alternative to relying on Google, Wikipedia, or vendor catalogs. Citation, a number of citations will usually take you to the oldest compound. Reviewers of journals will often tell you to use the old compound because it's so highly cited. It's often been superseded multiple times by much better chemical reagents, and chemical probes, which is why we need a resource to help biologists find the right tools to use. We need chemists to deliver those probes onto the portal. It's a resource, by the way. It's not a practical supply of compound. So expert review mechanisms are complemented by other resources like ProbeMiner and Drugs and Probes that can give you other alternative sources of advice, but maybe less acceptable perhaps to biologists. So how it works is researchers or curators on the portal identify compounds, they're put into the database, they're reviewed by experts, and if they pass, then they're recommended on the portal. And here are the types of compounds that we have of various types, mostly inhibitors. Here are the criteria, and you can find advice on the portal for the criteria. Here's the desirable elements as well, which is active control compounds, orthogonal controls, and inactive controls. Sorry, I should have said. There's a star system where 3 and 4 star are the more highly recommended. And once again, just to say, through the work of Bisan and Albert, who are also involved in the portal, it's complementary and interconnected to many of these other resources that are summarized here. So where are we? Metrics on the current status. We've got more than 600 compounds on the portal. We have a long way to go, of course. 250 of those are so-called historical compounds. Mostly you shouldn't use those. It's a place where you can go look for compounds, even if the reviewers of the journal tell you that you shouldn't use, and you can cite the portal for why. But there are 371 chemical probes acting on 300 protein targets right now. We got 170 expert reviewers that do the reviews, and there are nearly 900 reviews on the portal. Increasing activity, which was helped by a 1.5 million grant that we won from the Wellcome Trust to produce more probes and to get the message out to the community. Target class is Apache, lots of kinase inhibitors, lots of epigenetic inhibitors, not so good on some of these other areas. So we need to increase the number of probes. Generally, we need to increase the number of classes. The majority of the probes come from industry, nearly half. Academia involved so just over half, I think, or here you can see the numbers. It's not the vast majority, but it's a greater majority from industry. Academia are involved in nearly half. Many come from academic and industry partnerships. And regardless of the provenance of the compounds, they have a similar split in terms of the quality. You can see here, I won't go into the detail there, but this is a breakdown by where the probes are coming from, as taken from the authors on the original publication. Can be up to 5 countries involved in multiple groups in the authorship. Large numbers from 2 countries, no data, because this is a quick snapshot just for this talk, so we're trying to complete the data here. Of course, a larger number of investigators and countries are then involved in biologically profiling the probes once they're put out for use by biologists, and that's crucial. This is the new portal. We've been improving the portal under the Wellcome Grant. Here's the new, if you want to take a look, we've done a soft launch after beta testing of the portal. So the new enhanced portal is coming. The new site is now live before the full launch. You can go take a look. Please give us your feedback if you do. You can search and browse by the target or by compound. You can find useful information. You can check out the criteria that we use for these different types of probes. We've done major improvements under the hood, and the underlying database and the infrastructure has been massively improved, more user-friendly, faster to submit an online submission, automatic population of fields, for example. Please submit your ideas and your exciting probes and the feedback to help us populate the portal. Open for new probe submissions. Please join us. Here's an example of, I'm using this deliberately, of a compound that came by us. You're not allowed to, from us, you're not allowed to submit your own probe, so that it would be done by another independent group and reviewed independently. But this happens to be one that came from our lab. You can see the type of information that you get. You can see the SAB ratings for use in cells or in whole animals. You can see some information from that the SAB have commented when the paper was reviewed. This is not on the portal, but this is just to show you a compound I'm going to tell you about now, which is a highly selective and potent inhibitor for CDK8/19, as shown by kinome in vitro profiling, and also in the very nice paper by Cara Wells and colleagues from SGC, UNC, and Promega, where they had whole cell CDK engagement in live cells, target engagement in live cells, that highly selective. Only the two CDK8 and 19 kinases were engaged in a whole cell setting using that assay. So this is a fantastic collaboration between ourselves at ICR, Cardiff University, Trevor Dale's group, Merck in Darmstadt, funded by Cancer Research UK. Here, what we, in a phenotypic screen, we found this compound, this tri-substituted pyridine compound that you can see in a Wnt reporter screen. When we pulled out the target using the compound on a bead and pull out the target from cells, crucial targets turned out to be CDK8/19, which are part of the mediator complex. Crystal structure confirms the activity on the target with many other biophysical methods as well. And this is target engagement assays in cells showing that cells, when treated with this, inhibit the desired downstream consequences of inhibiting the target. So this is to illustrate the requirements that I showed you in the earlier slides. Mediators involved
Paul,
in.
I hate to rush you.
Need to finish quickly. Yeah,
okay, fantastic.
Sure. Yeah, well, quickly through. So basically, what we show here is if you knock out the target CDK8/19, you lose all the activity of the compound. So it's working on target. However, for cells in vitro, cells in a tumor, you need to inhibit both CDK8 and CDK19. So this is coming up to the end, Matt. Sorry for slightly overrunning. This is our open source project that we're doing with all these fantastic organizations funded by the Chordoma Foundation and the Mark Foundation. I have a passion for this because my mother died of this very rare tumor called chordoma. We worked previously with a group at the Broad and Baylor to show transcriptional CDK inhibitors block the expression of brachyury, which is the gene that causes this disease. And now we're trying to find, working with Ofer at SGC, working with David Drury and colleagues and Cara Wells at UNC, North Carolina, and our lab to find inhibitors, both binders and biochemical inhibitors, and also degraders of the brachyury target. So just my thoughts on chemistry inputs very quickly. Ambitious project, this. Chemical innovation important for PROTACs and molecular glues. Discovery high quality chemical probes requires close collaboration with chemists and biologists. Iterative design and make and test cycles are crucial, just as it is in a drug discovery project. So I took this from, I don't have the source, but I took it from the web from, I think it's from Wikipedia. But you can replace probe for drug. You can replace chemical probe for drug candidate. But some of these features like toxicity and so on, HERG and so on, are not required for an in vivo probe. But you do need to do a lot of SAR and a lot of chemistry to get, and biology to get to a probe. I just mentioned again, chemical probe requirements can be more demanding than that for a drug. Team science is required to accelerate progress. This is the fantastic team that works with us on the portal. This is the team that worked with us on the CDK8/19 project. Thanks to them. Thanks to you for attending this. Hope that was useful.
Thank you, Paul. That's fantastic. It's an amazing resource and an amazing open science story, the portal. So thank you for that. If anyone's got any questions for, please do put them in the Q&A before we finish. Then a few more minutes. I guess I'm just going to want to hand over to Cheryl to close things, but I guess it's been really inspiring to see such great examples of open science, accelerating research. And having seen some of these things, I mean, hopefully everyone gets the excitement about working in this way. And who would want to work in a closed way when you see such great projects like this? All right, I just wanted to invite Cheryl, to close the session and just express my sincere thanks to all our speakers today. Thank you for coming along.
Right. Well, thank you, Matt, and all the speakers. This was a great session. We've learned about a huge untapped, mostly untapped resource of chemistry educators and learners around the globe that could be brought into the working towards the goals in the open towards Target 2035. We've heard about the importance of educating beyond the chemistry community into the users of chemical probes through the Chemical Probe portal and the exciting new news of the launch of the new portal. So I look forward to all of you in these leadership positions in this area and your communities to coalescing towards the Chemical Probes initiative of Target 2035 and working together over the coming few years to try to get this going and accelerate. We really are going to need to accelerate the rate at which probes are discovered and disseminated. So thanks everyone, and we'll see you next month for the next webinar.